All right. I think we are ready for this first session. My name is Dita De Montis. I'm from Aarhus University, and I'm also a part of the NFF Center. And I'm chairing this session, and I have been really looking forward to hearing these three speakers. And the session is on scaling variant interpretation. And the first speaker is Ruth Lage, and Ruth is professor at the Novo Nordisk Foundation Center for Metabolic Research at the University of Copenhagen and the Novo Nordisk Foundation Center for Genomic Mechanisms of Disease at the Broad Institute. So welcome, Ruth.
Good morning. Thank you so much for having me, and thank you for the introduction, Dita. My talk is on the genetics of obesity, and more specifically in how we actually should curate our phenotype, in this case, obesity, to get to the next level of variant to function translation. So very briefly, my disclosures have no effect on anything I tell you. So no secrets here. Obesity is a traditional common disease, just like type 2 diabetes, cardiovascular disease. The environment has an important impact. Over the past 30, 40 years, the prevalence of obesity has doubled, tripled, and that's definitely driven by a changing environment. We eat more, we move less, but it's in particular people who are genetically susceptible who will respond to this environment and who will gain more than people who are not so much susceptible. So the heritability has been estimated to be 40 to 70%, which is considered, and again, very similar to other complex diseases. Maybe different from other complex diseases is that obesity is a very visible public disease, but I'm not going to talk about that today. But it's also a very early onset disease, unlike type 2 diabetes and cardiovascular disease. So that's when we think about prevention, that's something we need to take into account. Furthermore, it's also a disease that involves a lot of organs or tissues, not just the brain, not just the fat cell, but when it comes to it's like connecting obesity with comorbidities, many other tissues and organs are involved. When it comes to gene discovery, it's been actually a very successful pathway, I would say. Is like we started in 2007 with the discovery of FTO with only a sample size of 5,000 individuals. And then over time, we've identified more and more variants, mostly because we increased the sample size more and more. And many of these efforts have been driven by the giant consortiums, like headed by Joel Hirschhorn. And by now, we have more than 1,700 variants that have been identified, mostly associated with body mass index, or first identified for body mass index. A few have been identified for other adiposity-related phenotypes. And what we've learned in the context of the biology is that the brain is a central organ, probably affecting like our hunger, satiety, reward systems in the brain. The hippocampus, limbic systems are driving this whole process. You see here, this is a nice figure from 2015, but it is still the case. Not much has changed since then. But the variant to function translation has been very slow. FTO was the first discovered phenotype almost 20 years or 18 years now. You see a very nice signal. This is again from an older paper where the p-value is 10 to the minus 60. Now we're at like 10 to the minus 200. And initially we thought that FTO was the causal gene, and there was quite a few papers in Cell, in Nature Genetics that showed very credible results for FTO as a causal gene within the context of obesity. But soon after the discovery, researchers started to venture out. Like some thought it was RP-GRIP1L that was the causal gene because it's like encodes the basal body of the primary cilia, which we know from the Bardet-Biedl syndrome may be cause of obesity. But like others, including Melina's team, thought it was more IRX3 or even further down IRX5 that was the causal gene. So there was a little bit of discussion in the field, but then over time, things got clearer, and maybe this story got concluded with the paper in Science by the Marcelo Nobrego and Melina's team, where they indeed concluded that the signal here in FGO within neurons and pre-adipocytes seem to reach to IRX3 and IRX5, and that may be sort of the causal genes within this locus. And this is just an example to tell you, it's like it took them quite a few, it's like it took them about 14 years to come to that conclusion. And there's 1,700 variants actually that are waiting to be translated. Maybe there's like a dozen of variants that have been translated from variant to function within the context of obesity, but that's about it. Within my team, we've tried to approach the variant to function in a more it's like bioinformatic way, it's like a statistical way, because I don't do actually lab work. So Diane used, I think, about 8 approaches that were at the time that she was doing these analyses to do the gene prioritization. And then she counted how much the same gene was prioritized by the different approaches. So here summarized in this upset plot, for example, here you see that here are the different approaches that she considered for the gene prioritization. And you see that 86 genes were prioritized by 4 approaches, and could be any combination. 34 genes by 5, and 6 genes by 6. So that's not all approaches, but these are these 6 genes, and I won't go into the detail, but some of them are indeed like confirmed, or not necessarily confirmed, but also supported by animal studies that they may play a role in obesity context, and not always, they do not always point to the brain, but often also like adipocyte biology. And then another project that is in process, and that's also like a collaboration with Melina's team, like is Virginia. She's doing using a more multi-omics approach, where we go from SNP to BMI through maybe gene expression and proteomics data. So what she did is she combined all the publicly available eQTL data across multiple tissues. Sometimes she did a meta-analysis. She did the same for the proteomics data and used that in her analyses to basically align all the variants that we have. And I should have emphasized, these are the 500 variants that have been reported in the Yang et al. Paper, which is the latest giant paper. She prioritized 413 genes in 189 loci. Again, an upset plot, but the main message here is that, again, the brain remains the most, the tissue in which the most genes are being prioritized or seem to be playing a role. But other tissues are now also popping up. She then overlaid it. So here are her results based on the colocalization with eQTL and pQTL data. She overlaid that with the data from Diane right here, and then overlay that also with the results from CS2G. And you see there's 37 genes that are prioritized by all 3 methods. And now she focuses on these 37. Here are some of them to really summarize the data in a paper that will be coming out soon. So this is a plot that we use where we have a variant, the genes that are prioritized in which tissue. So it's just a lot of data to really summarize into one nice story. But that's coming up, because what's next is actually like the giant consortium has been working. So I haven't updated this plot since 2021, and that's not because I've been lazy, but it's mostly because there haven't been that many new discovery studies. The giant consortium has been working on a meta-analysis. Giant itself combined the data of 2.1 million individuals and then collaborate with 23andMe. So now we have a meta-analysis of 5.6 million people, and this is so now we have really 9,000 loci actually that we have identified, and the Manhattan plot looks like this. It's funny. So the significance threshold is right here, and like you ask, like sometimes it's like too much data, not necessarily helpful. So what can we do with this kind of data? We thought, like, let's start with the easy things. Instead of variant to function, let's just make a new genetic score. And we published that in the summer. It's a rule of lattice effort. So now we made a polygenic score that works actually really well. It's like better than the previously reported genetic scores. You see here the results in the European ancestry. Like, let's focus on the blue dot here. It's like the explained variance is now 17 0.5%. And we see actually it does pretty well in other ancestries, in Central South Asian, East Asian, Middle Eastern, at mixed American, but still not in populations of African ancestry. So that's definitely needs more work, and maybe a starting point of predominantly European data is not the way to go. The prediction of obesity has gotten better. So here are the results in BioMe, the Mount Sinai Biobank, Million Veterans Program, UK Biobank for obesity class 1, which is BMI over 30, then class 2 over 35, and class 3 is over 40. And focus on these results here, then you see that the area under the receiver operating curve is getting pretty good. So it's like 0.7 for just your regular obesity. And you have to keep in mind like that obesity is still 50% heritable, 50% or 50% genetic, 50% environment. So we'll never be perfect in predicting regular or common obesity. So 72 is pretty good. And as we go to more extreme obesity, we get better. And maybe that is because more extreme obesity is maybe even more genetic than just your common forms of obesity. And we see this already at a very young age. You see, it's like this is data from the ALSPAC study from 0 to 25 years. And we have 3 curves with the people who are in the lower decile of the polygenic score in red, the people in the top decile in blue, and then the average in yellow. And you see already at age 5 that genetic predisposition is coming through, and it puts you on a track throughout life to be heavier or leaner than the average, depending on where you are. Another thing we're doing right now is led by Victor, who's also here, is just building on the idea that genetics can support drug development. We're using the giant data to look in how many of the loci indeed provide support for for drug discovery. So this is very preliminary data. So out of the 9,000 loci, we prioritize 7,000 genes, and 700 of these genes actually are already the target of either approved drugs or drugs that are close to get onto the market, but not for weight loss. So the question is here, like, how many of these drugs that are approved or in the process of approval could be used for repurposing in the context of weight loss? And then among the new targets that are not currently, or among the new genes that are currently not a target for current drugs, how many could be new targets? And there we use the data from the Illuminating Druggable Genome Initiative. And here we see that again, about 800 genes are predicted to be druggable with high confidence. So maybe among those, there's maybe new anti-obesity targets. Of those 700, so most of them are antibody druggable, and 100 of those are small molecule druggable. Again, very preliminary data, but these are projects basically to avoid the real task, which is the variant to function, right? So because remember, I told you it's maybe a dozen of variants associated with body mass index that have been translated into function, but now we have 9,000. It's a little overwhelming. It's been taking a long time for us to really write up the story into a concise and meaningful paper. And maybe the real problem is not just the amount of data, but is also the phenotype. Some people have tried to shortcut the variant to function by focusing on rare variants. Here you see a few initiatives. The Giant Consortium here used the exome chip quite a while ago. It was a cheap way to get to the rare variants. We had 700,000 individuals find actually 3 variants that are truly rare, below the 1% level, including the 2 variants in the GIP receptor. These are coding variants. A paper by Regeneron that combined data of 600,000 people identified 16 genes, mostly focusing their paper on GPR75. We don't know yet whether that's a real drug target, but they are definitely working on that. And then there's a recent paper by the Cambridge group combining data of 400,000 individuals, and their paper mostly focused on bassoon. This is just to say that this seems an efficient way to go from variant to function within the obesity field, but you still need a lot of data for a few targets. So maybe the problem is not so much with the amount of data. It seems that we have a lot of data, but maybe the problem is really with the phenotype, body mass index. Everyone agrees that it's actually not a very good phenotype because it's pretty crude and it's far from the real biology. So in my group, we've really been focusing on curating the phenotypes, focusing on more refined adiposity traits. One study is focused on body composition, really, and tomorrow, Victor will talk about this. I think it's like an exciting study where we look at variants associated with muscle mass and fat mass in the same direction or in the opposite direction. He'll tell you more about that. I'll tell you a little more about the uncoupling of obesity from its comorbidities, and that's coming next. But we're also focusing on using PET-CT scan data from medical records of Copenhagen Hospital to quantify brown adipose tissue activity, but also other adiposity phenotypes. And that's very much in development as well. So we use AI to do the quantification. And we're also focusing on fat distribution. So obesity it's not only about too much adiposity, but also where exactly you will store it. And we also increasingly focusing on biomarkers, particularly with the proteomics becoming increasingly available. This promises to be an exciting field as well. So today I want to focus on this story, and actually it just got published last week in Nature Medicine, and is led by Nathalie Chamy, who's here as well today, and by J.J. Wang. So what we did is we did not do a case control analysis of healthy obese individuals versus unhealthy obese individuals, because there's a lot of discussion about that. But we looked at these phenotypes, body mass index, body fat percentage, and waist tip ratio on one hand, and these cardiometabolic risk factors on the other hand. So lipids, blood pressure, and glucose and insulin. And what we did is, so this story actually builds on a previous story where we use summary statistics. So here we use individual level data of mostly of the UK Biobank. So what we did is we take a distribution of body mass index. It's right skewed, but we made a standard normal distribution with a mean of 0, standard deviation of 1. And we did the same for all the traits. And also for the cardiometabolic traits. So each phenotype is now on the same distribution. And we made a new phenotype by subtracting one from the other. Say my body mass index is +3 on the BMI side, but -3 on the LDL side. So if you subtract one from the other, depending on which one you subtract, now I'm all of a sudden +6. So that becomes a whole new distribution. And now we did a GWAS on that new distribution. So if I'm +6, it means that I'm a healthy obese. I'm a person with healthy obesity because I have a high BMI and low LDL. If you're -6, you're the opposite. You may have a low body mass index, but you have a high LDL. And we did that for BMI and LDL, but we did that for all these possible combinations. So we created 24 new phenotypes. We did a GWAS across these 24 new phenotypes. We made sure that the signals we observed are co-localized, and we ended up with 205 new loci in 206, with 206 independent association signals. So just to confirm, we then made a score of these 206 loci, or 266 loci. So we have the uncoupling score, because we really look at how adiposity is uncoupled from these cardiometabolic risk factors. And we compared it with your typical unhealthy obesity score, you could say it. So red is the healthy score, blue is the unhealthy score. And you indeed see, like, if you do the association within the UK Biobank, there's no effect on height, but you quickly see that the difference is particularly visible in the VAT-SAT ratio, where if you have an unhealthy score, there's not so much an effect, and here's the null line. But if you have a healthy score, you actually have a favorable fat distribution. And you see the same for waist to hip ratio, where the unhealthy score is associated with a higher waist hip ratio, where the healthy score does not have too much an effect on fat distribution. It's more visible if you look at the risk factors, where here is the null line, and you see that the healthy score lies mostly on the inside on the negative side, so the protective side of this radar plot, meaning it's associated with lower blood pressure, lower glucose levels, lower HbA1c triglyceride, and of course higher HDL as expected. Whereas the unhealthy score is also as expected, lies on the outside. So this is just for confirming that what we find is indeed healthy obesity versus unhealthy obesity. We did a PheWAS within UK Biobank, and indeed we see like a nice confirmation that both scores are associated with increased obesity, but the unhealthy score is indeed associated with higher risk of type 2 diabetes, hypertension, and the healthy score has a protective effect. Of interest is, of course, that the association with other diseases, not the non-cardiometabolic diseases, that seem to be associated with weight-bearing diseases such as cellulitis, varicose veins, and so much. So it's of course a bit tricky to call it healthy and unhealthy, because in the end, the risks of the people with a high healthy obesity score, they're still associated with these diseases in the same way as the unhealthy score. We also confirmed these results in children, focus on the cardiometabolic risk factors. If children with a healthy score, and they're at around the age of 11 and 12, they already at that age, you see that they have a protective profile compared to children with a high score on the unhealthy score. If you look at the cardiometabolic, if you look at tissue enrichment analyses with a healthy variance, it's particularly the peripheral tissues that show up, adipose tissue, abdominal fat, and so on, but also cartilage, adrenal, stomach, whereas the unhealthy score, similar to the BMI score, is associated with the central nervous system. So clearly different biology that is being observed. We also look at the proteins. We know from the UK Biobank data that high body mass index is associated with high protein levels, probably because the bigger you are, the higher the levels. That makes sense, and that's shown in this data from Sun et al. So when we looked at the scores and their association with the UK Biobank proteins, the unhealthy score, which has some more power, is associated with more proteins. The unhealthy score is also associated with 337 proteins. 208 are associated are overlapping. Of the overlapping proteins, 85% are in the same direction. And this is what you see here. This is somewhat like there's 3 dimensions to this plot. On the right-hand side means higher adiposity, higher cardiometabolic disease for the red dots. But for the yellow dots, it means like a higher adiposity, lower cardiometabolic risk. And on the left-hand side, lower adiposity, lower cardiometabolic risk for the red dots again. And for the yellow dots, lower adiposity, higher cardiometabolic risk. So these 176 proteins are directionally consistent with the traditional adiposity proteins. Leptin, fatty acid binding proteins, you see them pop up in both at both plots at the same time, and the same like with some others on this side. So we call these proteins are adiposity driven. What about the 15% others? There's about 32 proteins that are actually going in the opposite direction. So they will show up here in the red dots, but here on the yellow dots, and on this side on the red dots, and here on the yellow dots. So, for example, growth hormone receptor, LDL receptor, ApoF, FGF21. Too much data to really go into the detail right now, but for us, it suggests that they're not driven by adiposity as such, because then they would lie on the same side, but now they lie on the other side. And what could explain here is that they're driven by the health status, the cardiometabolic health status. And then there's another, and then go a little faster, there's 129 proteins that only show up for the uncoupling, even though this uncoupling score has less power than the unhealthy score. We see adipoQ as expected to show up, lipoprotein lipase, myostatin. So there's a lot of interesting proteins that are showing up to be associated with either score, telling us more about the biology. Going a little faster, I showed you that we have one set of 206 genes that are associated, or variants that are associated with this healthy profile, but actually they also cluster within multiple subclusters. For example, here, it's a heat plot. These are the 266 variants associated body fat percentage and body mass index. And by definition, because that's how we discovered them, with a protective profile with any of these cardiometabolic risk factors. But you see they clusters. This cluster 3 is associated with lower body fat, lower blood pressure. Cluster 5 with lower glycemic traits. Cluster 6 with a healthier lipid profile. Cluster 1 is pretty complicated because it has a healthy profile for triglycerides, but unhealthy for glycemic traits. And then 4, 7, and 8, they're actually more globally associated with a healthier profile. So combining, and then we did a variant to function prioritization, bioinformatically overlaid with GTEx data, and then in collaboration with Melina, also using CS2G. And combining all that data and knowing the clusters in which these variants fall, and sort of having that more refined phenotype for each of these variants, we were able to prioritize some of these genes better than we could without that information. So here I'm just going through it, highlighting some of these genes. If any of these genes are familiar, like, please let me know, because we are now working on like the real variant to function. So even though this study was published, the study is not complete, and we're working with Melina's team and one postdoc in my group, Melina, Eleonora Manita, is here doing this real variant to function in the lab with Melina's team. This is just to say, and maybe to conclude, that it's like gene discovery is maybe complete, but we really need to work more on curating the phenotype to get the phenotypes closer to the biology, to facilitate and make it much easier to do the variant to function translation. So it's important to just not forget about the phenotype. So just to point out that the biology that we implicated with the genes that we have identified are the ones that we've identified before. But actually now with the more data, we strengthened these observations, and we actually identified a few new pathways that seem important in the variant to function translation. Just to say, like, identifying variants is not hard, but genetics may help elucidate the heterogeneity within obesity or any complex trait. We see multiple loci that uncouple adiposity from its comorbidities. Multiple mechanisms seem to be underlying, and by combining this better description of the phenotype with the variant to function translation, I think we may expedite the actual variant to biology observations. Very grateful to my team at CBMR, at the ICON Institute as well. Working with Melina has been a blessing, and it really has pushed me to the next level of the variant to function translation and going beyond the bioinformatics. And of course, also the giant consortium with Joel Hirschhorn leading it, and of course, the funding for this work. I'll finish here.
Thank you, Ruth, for a very, very interesting presentation. So are there any questions from the audience?
So a couple of questions, Ruth. So one, if you were to filter genes by just the number of genes that definitively have multiple variants that can be linked to the phenotypes of interest, does that help narrow the list? So I'll ask 3 quick questions. So that's question number 1. Question number 2 is, I never saw GLP-1 come up on any of your gene lists. So if you were to do a retrospective on GLP-1, where would it have fallen out kind of in this list? And then the final question is on FGF21, it looks like it goes in one direction and then the other direction. So it increases risk of adiposity and then decreases risk of cardiometabolic disease or cardiovascular disease. So if you were to perturb that, would you expect differential effects across those 2 phenotypes?
Right. Actually, I did the first question about filtering genes. What was exactly what you mean?
Multiple alleles at a locus to help whittle those down to small?
That's a good question. Actually, we haven't looked into that. But indeed, we are working on the variant of function in the lab with Melina. Don't know what the stage is at the moment, but yeah, waiting for the data. For GLP-1 receptor variants, it's very interesting with the 9 million, with the 5 million meta-analyses, it's starting to show up. It's a very nice peak on GLP-1 receptors, but it's the first time we see it in a meta-analysis show up. In adults, it's very intriguing, but a small 28,000 individuals meta-analyses in children, but in children before the age of 3, GLP-1 receptor variants are showing up there. And that's actually a phenotype. BMI before the age of 3 is not correlated with your adult phenotype. So it's very intriguing. We don't quite know what's happening. But then we need 5 million people to find it in adults. And then your third question, I already forgot. Yeah, it's true. It's like, and I didn't mention it here, but these are plasma proteins, and these are still very correlated phenotypes or correlated observations. We haven't looked really at the causality. Is FGF21 up as a causal or as a consequence or as a compensatory? It's like there's a few other of these proteins like LPL and myostatin that are in the direction that we didn't really hypothesize would be, but that may have to do with the fact that it's plasma proteins.
So our next speaker will be Jason Flannick. He's assistant professor at Pediatrics at Harvard Medical School and the Division of Genetics and Genomics at Boston Children's Hospital, and associate member of the Broad Institute at MIT and Harvard.
All right. Thank you for the invitation to speak here today, and thank you all for listening to me. I'm going to talk today about a method and resource for trying to understand mechanisms underlying GWAS results, and also we'll see rare diseases too. And I've called this a gene to variant approach because it comes at the V2F problem from a little bit of a different perspective. And that perspective comes from our group's work on genetics knowledge portals. If you're not familiar with our group, we build a lot of we work with a lot of communities to make their GWAS data and downstream work available through public knowledge portals. If you haven't checked out the Association to Function knowledge portal, I'd encourage you to do so. It has a lot of interesting results, I think, that's relevant to a lot of the things we'll talk about today. And this was a direct actual consequence of one of the earliest V2F initiatives here at the Broad. It was initially called the Variant to Function portal, and now it's funded by NHGRI to be the Association to Function knowledge portal. But our work in this area kind of goes back even further to the Accelerating Medicines Partnership for initially type 2 diabetes and subsequently common metabolic diseases. And this was a consortium about, I think, maybe 7 or 8 pharma partners now over its course with a bunch of academics and industry and nonprofits as well. And the goal was to make human genetic data more broadly accessible to catalyze drug discovery, primarily through prioritizing targets that had genetic support. And one of the first projects that we did actually as part of AMP-T2D was kind of a mini manual ad hoc V2F. This is way before it was called V2F. But we took all the type 2 diabetes genetics data that existed at the time. We kind of cross-referenced it with all these other experimental data. It was very kind of messy, but after about 6 or 7 months, we produced this report with a list of potential targets that we thought were, had very strong genetic support from the genetic data as well as auxiliary genomic data for type 2 diabetes. And what I think was notable to me about this activity was not that literally every single one of these genes was completely rejected by every pharma partner that we worked with, but that the reasons that they rejected them was not because the genetics data wasn't strong or they didn't believe that these had genetic support, but that there were a bunch of reasons that were specific to each partner that just ruled them out for maybe it wasn't competitive in the market, maybe they didn't have an assay, it wasn't an area of interest. So there were all these like very specific considerations that each partner had. They could have told me in advance, like, we're not going to be interested in that gene if you prioritize that. So that's kind of how I thought about this over the years, is that we typically think of the variant to function approach. I think Ruth just illustrated this. We have like 7,000 loci. How do we come up with technologies to map each one to, you know, what it's implicating functionally? That's very difficult to do. And also, a lot of these loci are not going to be of interest to a vast majority of people who are going to use them downstream. And instead, I think of the problem more on the lines of, can we answer the question that if a user or somebody, you know, not just pharma partners, but somebody who would be an experimental biologist, has a question about the potential role of a mechanism or gene in disease, what can we tell them about what human genetics says about it? And if you think about this is, you know, while it's flipping the question on its head, it's still using the same data. But you can imagine many cases where even if you haven't definitively mapped every single variant that informs on the function, and maybe even there isn't a genome-wide significant hit for a gene involved in this mechanism, you might still have information. You might have already even solved the problem. So kind of what I'm going to talk about today is how to answer a question like this, because I think a mechanism, we probably all mean different things when we describe a mechanism. But, you know, in my experience, most people have like a very narrow range of interests. You know, they have like an assay that they work in, they have investments that they've already done, they want like novelty, but not too much novelty. So a very complicated question like this, the question is, can we provide a resource that enables people to kind of, you know, address this? So I'm going to walk through a couple of steps of how we do this, and argue that I think with large language models and really the amount of GWAS data that we have across thousands of traits now, it's actually becoming possible to provide useful answers to this sort of question. All right, so starting from the ground up, I'm going to model a mechanism, right? So this is, I'm going to be intentionally very abstract and fuzzy. So I'm going to define a mechanism just to be a fuzzy set of genes. So I'm ignoring anything involving like pathway information, regulatory relationships. It's just, here's a bag of genes with some weights. So all the context for that mechanism, you know, like let's say insulin signaling is a mechanism. You might say the specific genes involved might vary by tissue, it might vary by context, disease state. Those are going to be encoded by completely different gene sets. So what this means is we might have like potentially an infinite number of mechanisms that are actually existing in the human body, right? And moreover, we don't observe any of these, right? We don't know actually the precise genes that are involved in any process that's really happening in the human body. What we do have are a bunch of experiments that we can do, which will produce a readout of genes, and that readout's going to be influenced by whatever mechanisms that experiment's targeting. So imagine you have like a differential expression experiment in the pancreas, people with type 1 diabetes versus healthy controls. You might hypothesize that genes involved in autoimmunity or insulin secretion or beta cell growth might somehow contribute to the readout of genes you get from that differential expression experiment, okay? And while we can observe like the true mechanisms, it's trivial, not trivial, but extremely easy to generate tons and tons and tons and tons of these readouts, right? In fact, there's like millions of them scattered across all these different biorepositories. I work with a colleague, Avi Mayan, actually at Mount Sinai with Ruth, who has built a ton of resources for doing this. And while these are biased and noisy and incomplete, you would assume that like the label of a gene set, you know, hopefully has something to do with the mechanism it's interrogating, right? So this, the genes involved in, you know, annotated as having abnormal adipocyte glucose uptake when you delete them in mice, hopefully inform on some mechanism that's relevant to that. So you can think of this giant matrix of like every gene by like every readout you have, everything's noisy, it's incomplete, it's biased. It's almost kind of like, if you think about it, like a GWAS tag SNP, right? So like the tag SNP is often not the actual causal SNP. Here, you might tag a mechanism with the gene set, but may not necessarily be the causal one, but this is still our input information. So what we're trying to do in the context of V2F is when GWAS is the readout. So GWAS is going to read out like, you know, between cases and controls, so however you define them, a set of variants that implicitly observe a readout of genes, okay? And we're trying to unpack which mechanisms are kind of contributing to that readout, okay? And our goal is to kind of go back from the GWAS results to infer the underlying mechanisms. So here's a statistical model as to how you can model this. So I'll build this up from the ground up going forward. So on the bottom level, we have a variable for every gene set in that library. So it could be 1 million of these, okay? And they have latent weights, the degree to which they contribute to the trait. So there's one model per trait, okay? So we don't know what these weights are. We do know the gene sets, and we know which genes are in them. And so we have those feed forward, and they add on the log odds scale to produce a probability of disease relevance for every gene. So we have 20,000 additional variables for genes, and those probabilities are also latent. We don't observe them, okay? We can put and then I'm kind of glossing over the details, but you put regularizing priors to kind of handle like the super large scale of like these number of gene sets. So what you do observe are the variant associations nearby each gene, okay? And so you need basically, this is the key, right? You need a model of how the genes, whether or not they're disease relevant, influence the associations you observe nearby, okay? So obviously this is a very hard problem. To some extent, variant to gene is like the whole thing we're trying to do here. But I would argue that while going from a specific variant to confidently implicating a gene is very hard, it's comparatively easier to, given knowledge of a gene being disease relevant, to look at all the variants nearby and model how their associations change. And so we have a postdoc in the lab, Alex Llamas, who's working on this. Actually, a lot of the work that's been done by people here at the Broad, Alkis Price, Ben Neal, in particular over the last decade, have really worked out how variant effects change in response to like annotations that are relevant to disease. And you can kind of incorporate genes into this model and like, again, lay out equations that are really just directly informed by the work that a lot of people here have done. I could spend like a whole talk on this. I'm not going to do that today because I want to more focus on kind of the mechanism inference here. And also this method is still a little slow to use as part of what I'm talking about here. So all the results I'm going to show today instead are a much simpler model of how variance effects change as a result of being near a disease-relevant gene. This is like a very, very simple, is a variant nearby the gene? How far is it? It's called human genetic evidence scores that we have a perspective on a couple years ago. Again, I'm not going to go into the details of this. I'm instead going to go into like kind of how you infer and what you get when you try to back down and calculate the genes and the mechanisms that underlie a GWAS. So again, not going through the details, you observe the GWAS variant associations. You can then, you want to infer the latent gene probabilities and mechanism/gene set weights, okay? You do that via a technique called Gibbs sampling. I'm not going to show the equations for that. And you output a completely joint posterior distribution over all 20,000 genes in every gene set. And by nature of and again, these priors we put on the gene sets, it's actually a very sparse. You kind of select out specific gene sets in a way that's much more interpretable. So what that looks like, I'm showing results for like a heart failure GWAS. You basically drop in the GWAS, it runs in about 20 minutes, and you get out. On the left table is showing kind of those gene level variables. So that leftmost column is those probabilities of disease relevance. And those are actually broken down on the log odds scale into effects coming down from the variance. Okay, so that's what's called GWAS log odds. That's how much evidence there is from the GWAS for that gene. And then bottom up, how much are coming from the gene sets as part of the model, right? Everything's being learned from the GWAS, though. There's no like pre-specification of the gene sets. On the right it's showing the top gene sets that are selected. And these are, remember, they're jointly fit. Okay, so they're all like jointly modeled as to how they contribute to gene probabilities, which is a distinct thing from fitting them one by one, which is done in most traditional enrichment analyses. And I'll return to that in a second. You can kind of see, like, I don't know how many of you know about heart failure. I don't know much about it, but at least the gene sets and the genes a priori, if you look these up, they all kind of make sense. Now, one thing I also want to note is that I kind of couched this in the language of inferring mechanisms from a GWAS, where you have the SNP data. But if you have direct estimates of gene probabilities, which you often get for a rare disease, for example, you can run the exact same model. It's just here that rather than you're just directly encoding the gene probabilities, and you can get that from like OrphaNet or the GenCC, and then you can run the exact same model in any rare disease, right? So this is like kind of a rare form of heart failure, cardiomyopathy. And obviously you can see from that known log odds, that's what we're hard coding these genes to have a 95% probability from the data, but you still infer the underlying gene sets. And it allows you to actually, and I'm not going to touch on this, but kind of embed these rare and common traits in this like kind of gene set space, which has a lot of interesting implications. So the method for doing this is called PIGIN, and the name for this is priors inferred from gene annotations, and that's because the way I usually talk about this in its initial motivation was not to infer mechanisms, but to just infer gene probabilities. And you can actually show that if you typically think of genetic support as the probability of genes involved in disease given its associations nearby, if you augment that to also consider its annotations, and then you fit that jointly, you can kind of decompose like the estimate into a prior that comes from the annotations that you learn from the data, and then direct evidence, typically a Bayes factor for those who know Bayesian statistics, coming from the direct data. There's another interpretation, if you're familiar with the POPs method. You can actually show that if you kind of do a couple of modifications to kind of approximate or use heuristics to kind of truncate this model, you can show that you get the POPs method. So this is kind of just a generalization of POPs to be probabilistic and directly fold in, both direct evidence and also associations alongside the annotations, which allows you to kind of, have a justification of what POPs is doing when it's prioritizing genes at a locus. But the interpretation I like the most actually is thinking about a new concept of genetic support, which I call indirect genetic support. So we typically think of support for a gene coming on the left just from the variants nearby the gene. But on the right, you can kind of see that support also comes from mechanisms that the gene is involved in that you infer are relevant to the trait. And that's from other genes across the genome that kind of implicate the mechanism. So your support for a gene actually comes from the associations for every gene in the genome with like the mechanisms that they share, kind of providing that link as to how that information flows. So the way I think you can think about genes that are relevant to a trait is actually on an axis of 2 dimensions, right? So on the y-axis is how much GWAS association evidence there is for a trait gene, and on the right, the x-axis is kind of how much information there is from just from the annotations. I'll have more to say about this momentarily. I'm not going to go through too much of validation for this. We've done some extensive validation. I'm just going to reference one, which is this concept of relative success. Eric Minnickel and others here at the Broad wrote this paper a year ago, and the headline finding was basically if you have a gene and it's near a published GWAS association or a published rare disease gene, it's like 2.6 times more likely to succeed as a drug target. And if you kind of we worked with Eric to do the exact same calculations, and what you see is for both direct and indirect support, as it increases, remember this on continuous scale, you have it for every gene in the genome. As you go from no support up to a very large number, you get a significant increase in relative success. These appear additive, so for whatever threshold you have for direct support, adding indirect increases it, vice versa. This is shown from just like all of the annotations, but we've also done this with like a time limited where you only look at approval after the date of which the annotations were developed. And putting it all together, you have an uptick that, relative to like the 2.6-fold increase from like, is it nearby a published GWAS hit? You get a shift up when you look at like the comparable, like full probabilities. And also, of course, a much larger number of targets that have support because, you have a lot of targets that aren't near GWAS hits, but have suggestive hits and also all annotations that we're inferring. So we're still working on publishing this method, but we've applied it to 7,000 traits. You don't actually even need full summary statistics to apply this. You can run it just on a list of known curated SNPs from the GWAS catalog. You are welcome to browse this it's not publicly linked from any of our portals, but if you just put /PIGIN at the end of it, you can type in any phenotype or gene and get a sense of how it's working. And if you have any thoughts of this, please, I'd love to hear from you as we're kind of working through how it might be of interest. Just to kind of walk you through like some of the basic things you can do. You can type in any trait, you get this plot I was showing. I think I switched to coronary artery disease rather than heart failure. But, there's a lot of information in these relationships, right? So if you expand any dot on the plot, you can see like, the locus zoom plot that's in this case contributing to this direct support log odds of 4.26, which is like an 80% probability. And also all the gene sets that were learned from the GWAS to actually support this, right? So this is like a gene SCARB1 that's kind of like in the upper right-hand corner of this plot. So these are like these are all going to be your completely well-known genes. But you can, depending on where you go in this plot, depending on your interest of novelty versus like associations, like things in this middle are slightly more novel, like just picking out one, like an example, TSPO, you'll see kind of where there's not a genomide significant variant nearby it. There's one that's like near genomide significant. But when you combine it with all these annotations for cholesterol metabolism, it bumps it up to have like a probability that might be higher than a gene that might be an effector, or that gene that's not clearly an effector in like a known locus. One thing I can't talk about, but I think is interesting perspective, is that if you flip this around and look at genes, you actually get these incredibly dense fingerprints, right? So you can almost just trivially tell from, across all these traits for this gene that it's involved in like heart muscle function. So it causes you to think about, is the real thing, I take a GWAS and define the phenotype and then I get the function of the gene, or do I just like take the gene and look across every trait and infer the function? And like actually an LMM is really good at guessing just based on the phenotypes what the function of the gene is. And that's before even getting that you have these two axes, right? So like the traits that have both direct and indirect support are where you have a very clear coherent biology, but there are genes that like, I saw this on George Church's slide, where there's like super well-known annotations, but no associations and kind of the inverse. So you have these complicated patterns of almost like these embeddings of these genes that I think tell you a lot about function. But I want to return kind of the remaining 5 minutes on back to this question of mechanisms for a GWAS. So if you kind of look at the gene sets that are at the top of the model, this is for, again, coronary artery disease, you can kind of tell by their labels that they're picking out different mechanisms, right? So like the cholesterol metabolism gene set looks like it's probably a different thing than the disorganized myocardium. So that one's a wiki pathway for cholesterol metabolism, the second's a mouse phenotype. This is very different from traditional enrichment, right? Because remember, we're fitting this jointly. So if you just sort by the marginal effects, you get like nothing but lipid traits, which is why, it's kind of similar to BMI, where you just see all the brain stuff at the top. That's because those are marginal enrichments in a lot of cases. And when you fit it jointly, you start to decompose into these orthogonal different mechanisms. And like, if you expand these and see the genes, like, they are completely different genes. So you're kind of segmenting these genes for the GWAS. But the issue is that gene sets, as I mentioned, are not mechanisms, right? They're tags you can think of, noisy, incomplete, biased tags for true mechanisms. And so here's where, though, if you model it like this, right? So imagine you have a matrix, I'm calling the gene matrix. That's the true mechanism matrix, as I defined it. Those are latent, you don't observe them. And then you model the gene set matrix as readouts of those latent mechanisms, okay? So we don't observe those, but we observe this gene by gene set matrix. And if you model this, you can show if like these are Poisson like distributions for the entry. It's a little messy. There's probably a better way to do it. You can use non-negative matrix factorization to decompose the gene by gene set matrix and get the underlying latent inferences. Okay, and it's easier to understand when you see the data. So for example, you run the heart failure results and you get like a factor like this that loads on these different gene sets. Now with an LMM, just based on the gene set labels, you ask it, what do you think this is doing? And it can give you a little pithy thing, okay? So yeah, you're kind of condensing, all of these tag gene sets on the right for, going back to heart failure, into higher order mechanisms, okay? And you can tell by kind of like the heart failure hypertrophy is probably picking up a different mechanism than liver lipid abnormalities and et cetera. To give you an example of what they look like for each latent mechanism, you have the list of genes, and those have loadings. And then you have, so this is kind of that original definition of a mechanism I define. And then you have the loadings across all the gene sets. So those are your annotations. It's what allows you to interpret what the mechanism is doing. That's for heart failure hypertrophy. This is for liver lipid. You can tell, obviously, very different gene sets and different genes. Again, they're all labeled just based on the gene sets, but if you take the genes in them and then you cross-reference them with like their PHWASs, you get what you would expect in most cases. So on the top, all those genes, this is independent data, right? Like associate with a bunch of lipid traits from other GWAS, and on the bottom, the heart development abnormality genes associate more with heart structure traits, right? So ideally, you'd probably build a joint model that incorporates these patterns of traits like Ruth was showing. But like, this is kind of illustrating that even with the bias and the noisiness of annotations, they're able to pick out stuff that you can expect to see from the trait patterns. It's actually, I really like browsing these. I encourage you to type in your favorite trait. There's some really intuitive examples, like, for waist tip ratio, it's a common marker of, body fat distribution. you get mechanisms for like the metabolic syndrome, which actually separates for like insulin hyperglycemia versus dyslipidemia, but also mechanisms like skeletal development, which of course would also be relevant to waist-to ratio. All right, so in the final 2 minutes, I want to return to this question, right? So this is a really complicated question that is not directly obvious how you can just map it to what I just showed you. But LMMs are capable of parsing this and actually doing a very good job of inferring what the user wants with whatever they want to type in. But I'm sure most of us are actually using LMMs for this purpose, but we're all aware they're only searching public data sets. They're searching, text, qualitative assertions, maybe even not even searching stuff and kind of informing using its training to answer. The way you typically get around that is through a process called retrieval augmented generation, where the LMM will fetch data from like a vector database or a knowledge graph, and you encode your data in that, and then you pass that into the prompt, and then it can kind of like add additional data to its prompt. But this is like a very difficult thing to scale. You need to kind of like manually take a GWAS and like extract all these assertions. So here now, though, you can think of this as like what I just showed you is this very high dimensional tensor, right? Where you have like phenotypes by genes, by mechanisms, and there's like a number for every single entry in the matrix that's all completely uniformly inferred, right? Using human genetics to calibrate everything ultimately. And it's still very complex. So like this is just like a segment of that. Like, here the squares are like the mechanisms and like the diamonds are like traits and the circles are genes. So it's really hard for us to interpret. But for an AI agent, where you make this available, now the successor to RAG is called MCP, and you give it access to other tools like internet search as well as its own training, it's actually able to parse these queries and actually retrieve the information we've kind of embedded and extracted from GWAS. And I have to stop here and not really give more details on like kind of the next step for this, but just kind of conclude with an illustration that if you don't even build a real agent, but you just give it a custom GPT on ChatGPT, you can see at the top, I just pasted in that query, and you can see it kind of like infers what you want and knows what filters to apply to like these combined indirect and indirect support based on prompt engineering. It can then contextualize that with like novelty, druggability, the competitive landscape, all of those things it knows pretty well, and actually arrive at like, a nice filter, like helping you navigate this data, and again, it's because, it maybe can't search GWAS SNPs directly, but it can search these higher order relationships. So I guess, I don't know if Melina, you're going to ask me, but if a sentence I would say to summarize for V2F is that, well, I think the problem of like every single association can we map it to function is hard. I think the problem of for a given function, do I have insights from GWAS that help me make better decisions in the real world? I think there's an enormous amount of structure already there. And I'm actually continuously amazed every time I run these methods how much GWAS actually detects at that statistical level. If you're interested in learning more, please contact me. We're working on these agents, and hopefully on our knowledge portals, you'll see them soon. And you can also contact me at our lab webpage or also our broader knowledge portal team. And thank you. I'll take questions.
Thank you very much for an interesting talk, and I would definitely personally be very interested in looking into your portal. So any questions from the audience?
Hi, Jason. I was curious, the linkages which are in linkage disequilibrium, when you put your indirect support, if a linkage is acting in one direction in one case and in another direction in another case, how does that show up on your gene scores?
Are you talking about SNP linkages, or are you talking
SNP
about
linkages.
like the
SNP linkages?
So that would all be encoded in that, I kind of glossed over it, but that mapping of given a gene knowledge of whether it's disease relevant, what SNPs are regulating that gene. And you would do that disease by disease, and like you would use like the SNP to gene and all the work that everyone's being done here. All that has to be encoded in that model, and that can get quite sophisticated, which is why I didn't talk about it today. But the answer is you'd fit that separately right now for each trait. But ultimately, you would want to fit a joint model of like every disease at the same time, whereas like knowledge of a causal variant for one gene would inform that it's causal for another related disease. But that's computationally intensive. So we're kind of doing that post hoc right now.
Thank you.
questions?
Yeah.
We discussed a lot, so if there's anyone else can go. Great work. I love it, Jason, as you know. I wonder about the definition and validation of mechanism itself, which, as you say and acknowledge, is massively noisy, particularly when you draw from gene sets as a prior or proxy for mechanisms itself, and particularly when we're talking about complex diseases.
Right.
So what are you thinking about the next steps of improving the definition of mechanism itself? And related to this, maybe a separate second question is looking at PHEVAs, jointly modeling phenome association studies for a particular target and drawing conclusions from that is postulated across the community currently. And I think it's very helpful to come to prioritization of cell types, tissues, maybe cell states. I would argue, though, that the space of physiological traits cataloged in biobanks is limited to go down to cellular mechanisms that are disease-driving.
Right. So, okay. I think you're correct that, so I define an experimental readout of a mechanism, and then I use the current gene sets as like kind of the state of what we are. But there's no reason like your readout has to be a biased curated thing, right? Your CRISPR screens and lipos, all those are actual features. So I think you could envision this almost as like a fine mapping. Again, this analogy of like the current mechanisms are tagged SNPs, right? The initial HapMap2 is very biased. So now can you like fill that out with like more fine mapping of those mechanisms, for example? I think second, like, yes, right now we are not really thinking right now about how to model more details of what's going on within a mechanism beyond the group of genes. The hope here would be that this would kind of get you at just the area, like a set of genes, and then that would narrow your search space, and you're ultimately going to have to do more experiments. The PHEVAS approach, the third thing, what I found really fascinating is you can take like a Mendelian disease gene, just run it based on the genome-wide association studies, map through this method, give an LMM like just the pattern of associations, and it can guess the Mendelian disease like much more significantly above chance. So what it tells me is that when you look across enough traits, you can get at like very, very, very specific phenotypes. And you don't maybe
that. Yeah.
And you may not need to necessarily like be as worried about finding people with that phenotype in a biobank, if that makes sense.
It makes complete sense. And I completely believe that it's powerful to predict the phenotype. What I wonder about is the mechanism, mechanism driven by, at the cellular level, is this,
So I can, so like I just ran a quick test yesterday, like for PLIN1, right, which is this rare disease gene. It's involved in adipose expand, right? So you give it just the PHEVAS plot. You say, LMM, what function do you think this is? And it guesses adipose expansion, even though there's no GBOS hits for it. So the question is like, is that a useful definition of mechanism? I don't know. There's a resolution at which you're going to run out. But I think the answer is, can you at least narrow it enough and then like fine map, quote unquote, within that space to fill it out more? It's kind of how I'm thinking about it. But yeah, there's no substitute for doing experiments, obviously. Yeah. All right.